We are going to be focusing now on the mechanism of how the bile salts and the pancreatic enzymes become secreted. All right. If you begin before, the bolus enters into the stomach, through which sphincter, the cardiac sphincter or lower esophageal sphincter, comes into the stomach. There's a high presence of acid. Acid begins to break down these molecules. Certain enzymes also help further this breakdown. You have your pepsins and your lipases. Pepsin breaking down protein, lipases breaking down fats, coming in, and now they go through this sphincter. So what is that sphincter called again that connects the stomach to the small intestine? The pyloric sphincter. And the pyloric sphincter allows for this new material now known as chyme to enter the small intestine. What is the name of the primary region of the small intestine? What was the name of the first region? So it's called the duodenum. So the first region is called the duodenum. In the duodenum, now you have this chyme. Is this high pH or low pH? Very low pH because there's a lot of acid. This stimulates cells, specifically known as the enteroendocrine cells of the duodenum, to secrete certain hormones. And these hormones are called secretin and cholecytokinin, or we can refer to it as CCK. So entero cells secrete CCK and secretin into the bloodstream. Now you have these hormones that are supposed to help further enhance digestion and breakdown in the duodenum of the cell. Now, these two hormones, when they enter the bloodstream, they go to their respective targets. CCK and secretin migrate to the pancreas, liver, and the gallbladder. And now what do? So, secretin's main function is it migrates to the pancreas to release bicarbonate. So you have secretin going to the pancreas. And remember, pancreas has both an endocrine and an exocrine function. So secretin is taking advantage of the exocrine function of the pancreas, releasing bicarbonate, HCO3. Is that a base or an acid? It's a base, right? Because you want to neutralize it. And it also migrates to the liver to allow for the production of more bile. So increase bile production by the liver. That's very important. CCK comes in and does twofold. It goes to the specific cells in the pancreas that are responsible. They have these exocrine cells, this duct called the acinar cells, and they begin to secrete all the zymogens. So zymogen are all of these inactive proteins, proteases, lipases, amylases. And so these specific digestive enzymes that are secreted from the CCK, stimulated from the CCK from the pancreas, migrate all into the duodenum of the small intestine. Another function of the CCK is also increasing bile production with liver, so they work together synergistically with secretin, but also increasing the contractions of the gallbladder to increase bile secretion, right? So we said, where is bile made? Bile is made in the liver, but it's stored in the gallbladder. CCK stimulates the gallbladder to contract and release bile. So it also increases bile production by the liver, but another main function is to contract the gallbladder to release bile salts into the where? Into the duodenum. And so now you have these two specific hormones. Secretin also plays a role in inhibiting the peristalsis of the chyme to come in. Also, secretin and CCK work together to decrease the parietal cells in the stomach to stop production of hydrochloric acid. And so when these hormones enter the duodenum now we can begin the further breakdown of these molecules, right? And so what's really important when we talk about this is that during the cephalic and the gastric phase, these reactions are inhibited. So while you're, digesting or, salivating, these reactions are actually inhibited. Not until the chyme entering the duodenum and stimulating these enteroendocrine cells, right? These hormones actually are never really released. And so during the cephalic and the gastric phase, what nerve is usually being active during that time? What specific nerve? Do you remember? It's called the vagus nerve. So the vagus nerve plays sort of an inhibitory response on the secretion of CCK and secretin until the chyme actually enters the duodenum.